I'll go ahead and get things started, and hopefully more folks will come in while I'm talking for the introduction. Welcome everybody to the first FRAXA Fragile X webinar. We feel an acute need for a scientific meeting since our Gordon Research Conference got canceled this summer. And so we're kicking it off today with an exciting symposium talking about brain organoids and therapeutic development for Fragile X and other rare diseases, and featuring Drs. Alysson Muotri and Fabio Tucci. I just want to note that everybody will be muted during the presentations, but we will have questions at the end. And the way to initiate a question is to use the chat feature, which is down at the bottom of your Zoom screen. Well, I'll go ahead and get things started by introducing our first speaker. Dr. Alysson Muotri is a professor at the University of California, San Diego, and a very well-known stem cell researcher. He's been one of the pioneers in the use of organoids for the study of developmental disorders. And he's going to be talking about that subject for you today. So without further ado, Dr. Muotri.
Thank you, Michael. Thank you all, the organization, to put this together. I think it's a pleasure to be talking to you, explaining a little bit of what we do in the lab, and to team up with Fabio on our research on Fragile X. So let's see if I can start sharing my screen here. All right. So what I decided to do is to give an overview of the technology and some of the projects that we have going on in the lab. And then Fabio will continue more focus on our efforts on Fragile X, which might be more relevant for this audience here. So my lab has been using stem cells as a model for neurological conditions for several years now. And we take advantage of stem cells because we think it's a powerful tool to recapitulate early stages of neurodevelopment. And the reason why we do that is because we need to understand what happens at these very early stages of the development, what happens in uterus, in the embryo. And this is a hard question for scientists because we really don't have access to experimental access to what happens in uterus. Instead, we have to rely on indirect way that is not on the best definition. For example, ultrasound. You cannot actually measure when the first neuron fire, how the first network start to wire, things like that. And we know, especially for neurodevelopmental disorders, that this first wiring in the brain is really important. So if you really want to understand the condition, you really need to go back to these very early stages. So we think that embryonic or pluripotent stem cells give us the opportunity to really go and mimic that developmental stage in the lab, in the controlled environment in the lab. So I just realized we have to click here. And you guys probably heard through the media that we grow mini brains in a dish. And I'll just start by showing this picture that exactly not what we have in the lab. So I personally don't like the name mini brain because it gives the impression that we actually have a miniaturized tissue in our culture. But this is, I'll show you exactly how it looks like. It is not like that. And even this technology has important limitations. So before I move on, I would like to highlight some of the limitations of this technology. So you keep this in mind when I describe some of the results that we have. First of all, I mean, most of these organoids, they contain mature cells. These are neurons that are not mature enough to start firing spontaneously. These organoids are not vascularized. So that's another reason why they cannot actually grow bigger. They will reach out about 0.5 centimeter in diameter. and they actually stop growing. Not all cell types are represented. We have 86 billion neurons in the adult human brain, and way more cell types that are not neurons in there. So the organoids contain about 2.5 million neurons. So it's just like a tiny fraction that we have. We don't know if we are growing them in the ideal culture conditions because we don't exactly know how the embryo does. So we try to mimic based on our limited understanding of embryology. And perhaps more importantly, we don't know if this technology is translational. If what we learn in a dish can actually be applied to the human brain. These are early stages of this technology, so we have room to improve. But nonetheless, my lab has been optimizing protocols to create brain-specific organoids for a long time, and this is one of the protocols that we optimize for the cortex. We start with single cells. So these are dissociated pluripotent stem cells that has the potency to be driven to a specific lineage. So we use dual-SMAD inhibitors, very transient exposure to those molecules, so we can neuralize those cells. And then by adding some specific growth factors, you can induce the propagation of these neuroprogenitor cells. And by removing the growth factors, you let them differentiate, and they do it by themselves. They self-organize and differentiate into neurons or the different subtypes of neurons that you can find in the cortex. So the beauty of that is that this is a three-dimensional organization. And again, I mean, it's all genetically encoded. So the cells do it by themselves. And we can keep these organoids alive for several years in the lab, which is also a good thing. Another advantage is that in a single batch of organoids, you don't have one or two. You can actually create organoids by hundreds, if not thousands. So this is Isabel, a postdoc in the lab, showing a plate full of these organoids. Each one of these white dots that you can actually see it with naked eye, it's one organoid coming from one batch. And because you can differentiate these organoids from iPSCs or induced pluripotent stem cells, these organoids capture all the genomic information from the person that they were derived from. The way they self-organize is very similar to the way the brain does, at least to our understanding. So it's from inside out. This is just a representation of the migration of the cells in a 2D. You plate one of these organoids. On the top, you have the center of the organoid, and you can see that some of these cells are migrating out. They use cables. These are radioglia cells that provide processes that guides the migration of these progenitor cells from inside to outside. As they migrate out, they start to differentiate and form the cortical plate. So this is very similar to what happens in human neurodevelopment. And if you do one cross-staining in one of these organoids, what you're going to see is a ventricular zone surrounded by the progenitor cells. And in some of these radioglia cells, you can see the processes in white here, where the progenitor cells will send newborn neurons to form the cortical plate. So as these organoids mature, the definition of these structures becomes better and better, at least in our hands, in our protocol. If we dissociate one of these protocols at a specific time point, we can identify the different cell types using single cell transcriptome analysis. So this is just a snapshot at one of the organoids at 4 months of age. You can see that you have the vast majority of the neurons are glutamatergic or excitatory neurons. There are a cluster of GABAergic inhibitory neurons already present. This is more or less when the astrocytes or the glia cells start to pick up. So you have a representation of the astrocytes there as well. And we continue to have proliferation from the NPCs or neuroprogenitor cells that will continue to produce more and more neurons over time. So it's a very dynamic structure. This representation works at this time point. If you look earlier, most likely you're going to have only neuroprogenitor cells and some excitatory neurons. The inhibitory neurons and the astrocytes will appear later on. And if you keep them even more mature, you start to see the definition of the subtype of cortex. Different cortical layer markers will appear as well. So now that we have structures like that, we start using to model neurological conditions. I always like to show our work with the Zika virus, because it's one of the most striking examples of the uses of this technology. I'm from Brazil, and I was involved in Zika virus research and trying to find the causative agent for the microcephaly outbreak. The Zika was one of the candidates, but there was no experimental proof. So what we did was to expose these brain organoids to the Zika virus so we could see how the Zika infected the organoids and was able to kill those progenitor cells, reducing the thickness of the cortical layer. So that was a very clear example of how we can mimic the condition in a dish. But even more importantly, since we showed that the Zika virus can attack these neuroprogenitor cells, we could screen for drugs that were able to block the viral replication. And just 2 years after the outbreak, we were able to find an antiviral, or to repurpose an antiviral for hepatitis C for the Zika outbreak. This is sofosbuvir, can not only block the viral replication, but also block the vertical transmission. So when mom is infected and if takes this drug, the fetus would be protected. So we never needed to use these drugs because the outbreak in Brazil was controlled by controlling the mosquito population. But in a case of a new outbreak, we already have the drug available. So that's one example how we can speed it up drug discovery. It's important to highlight that we did some experiments in mouse in parallel to see if the Zika virus would cause the same birth defects in mouse. And it was really hard to see, mainly because the developmental stage of the mouse brain is really fast. So it takes about 24 days to have like a fully formed mouse brain, whereas in humans, it takes 9 months. So this is slow developmental stage was crucial for us to see how the Zika virus actually act in the brain and find drugs that were able to block it. So that's one example. So another example that we are quite happy with that is when we start modeling Aicardi-Gutierrez syndrome. This is a rare syndrome. There's about 200 families in US, and it's caused by mutations in a gene called TREX1. The kids are born with severe microcephaly as well. So the neurons just can't survive, they just die. And it's important that this gene, it's a single cDNA, there is no intron in the gene. And when mutated in mouse, there is no neurological problem. So we thought that using the human model here would be like a great advantage to the understanding of this condition. And what we figured out is that there is an intrinsic innate immune activation coming from the astrocytes that actually releases high levels of interferon response that kills the neurons. So by using this model, we're able to test the specific drugs that can block this innate immunological reaction in the astrocytes, preserving the neurons. And we were able to use that to use anti-HIV drugs to do that, as well as interferon blockers. So that was enough evidence to start running clinical trials. So right now, I have a collaborator in Europe, Yannick Kram, who has been using this strategy to reduce the levels of interferon in kids with AGS. So very translatable, didn't need any animal model to move from the tissue culture actually to a clinical trial. So it's one of the early examples of how this technology was able to speed up drug discovery as well. So this is some of the data in our brain organoids derived from AGS people. So you can see that the organoids, as the time passed, the size of the organoids really reduced compared to controls. This is because the neurons start to die quite prematurely. But if you treat these organoids with these specific drugs, you can rescue that. So the neurons are now preserved. Of course, you cannot restore the lost neurons, but you can block the progression of the disease. So that's our strategy now for this condition. All right, so this is all good, but there are several conditions where you don't see neurodegeneration, you don't see neurons dying, but you do have problems in the network from the formation. And I told you that the neurons in the organoids, at least in the past protocols, were quite immature, they were not really active. So we spend lots of time to find better physiological conditions to grow these organoids in conditions where the neurons will start to fire. And so in the beginning, we're very skeptical that this could happen, but I'll show you some very interesting data. The way we measure the electrical activity on these organoids is by plating them on dishes that contains multi-electrode arrays. These are electrodes that are printed in the bottom of the dishes, and the organoid sits on top. So as the organoids fire, you can capture this activity from the electrodes that are in the bottom of the organoids. And the idea was to start measuring them over time. And the reason why we were skeptical is that because most of the data in vitro so far coming from mouse primary neurons, rat primary neurons, human primary neurons isolated from postmortem tissues, they all show that you can never cross the 5 Hertz threshold. So this is a threshold of brain activity. So all the in vitro studies are below this 5 Hertz threshold. And the in vivo tissue, such as the mouse cortex, it fires about 18 Hertz. In a mouse primate brain, it's about 20 Hertz. So, I mean, it's very far from the in vitro system. And the reason we thought initially was, well, you cannot keep these in vitro cells alive for that long, but the organoids you could. But it's not all the organoids, so you have to really optimize the conditions for them to fire. What you have here, it's a curve of the recording that we perform weekly for 40 weeks. And you can actually see that there is a slow growth, but it reaches almost like an exponential curve, reaching to these 20 Hertz after 9 months or 40 weeks of culture. So this was unprecedented in tissue culture. And if you have that much activity, you start generating what we call brain waves. So these are the oscillatory waves that you can detect by EEG. So in the human brain, you have so much activity that even by placing electrodes outside the skull, you can still detect these brain waves. So we ask ourselves, can the organoids generate these sophisticated oscillatory waves that we see in the human brain? And the answer is surprisingly yes. And they start to oscillate when they are about 4 months. You can see some peaks. When they reach about 6 months of age, the oscillatory waves are quite stereotyped and synchronized. But if you wait a little bit longer, if you pass 8 months, they become highly complex. And this is exactly what you'd expect for a complex system such as the cortex. And not only that, you can actually compare these brain waves with premature human brains, since some of these premature babies will have EEG recordings to monitor their activity. So we could design a machine learning algorithm or a regression model to compare some of the parameters from the EEGs to the brain organoids. And this is unbiased, so the machine doesn't know if it's looking at an organoid or the real human brain. And the data strongly suggests that we do have, after 25 weeks, a very nice correlation between the oscillatory pattern that we see in the organoids with the human brain. And the trajectory, at least for the time that we recorded, looks really good. So from 25 to 9 months of age, the progression of these brain waves follow what is expected for the human brain. We don't have data before 25 weeks on the human brain to feed our regression model. And that's why what you have here in orange is a poor correlation, just because the machine never learned how the human brain fires at before 25 weeks of age. So What we are doing with these brain organoids that contains activity. So we are looking for opportunities to compare these organoids derived from patient cells with the actual human brain. So this is one initiative, is a clinical trial for autism. This is severe autism. It's a clinical trial to test the efficacy of cannabidiol or CBD. And there are 30 participants. We are running this clinical trial here at UC San Diego. And from all these participants, we're going to generate their own brain organoids, and we're going to be treating these organoids with CBD as well and analyzing their brain waves. So we have this unique opportunity to compare how the human brain behaves before and after the drug, and how the organoids behave before and after the drug. So the ultimate goal is to see or to challenge these organoids to see if they can predict the outcome of the clinical trial. If that's the case, that would be fantastic. We will be able to run parallel in vitro clinical trials to inform the real clinical trials in person. That would be a way to speed up and save lots of money by reducing or choosing the population that would be more responsive. So, but there are lots of other conditions that there is no specific treatments or cure. So we are looking into some of these conditions as well. To do that, we need to really optimize the way we grow these brain organoids. So we continue to develop better ways to decrease the cost and to move to a more high-throughput fashion. So there are protocols now that allow us to do things like that. And we also have a pipeline for several diseases, several conditions, especially the genetic ones. These are the ones that we're more familiar. So we can create isogenic cells using a single genetic background where we manipulate the genome to carry all these genetic alterations. And then we characterize phenotypically how the cells behave in the organoids, and we use that to test specific drugs or for gene therapy in the ones that are more target for gene therapy. So again, I mean, the goal is really to speed up drug delivery and gene therapy strategies. This is one example of one condition where we could do a medium throughput assay with a little bit more than 1,000 of drugs, and we screen for drugs that can correct or can alter the electrophysiological pattern of these spheres. So we are looking for calcium imaging here just because we can do it with imaging. And we create these plots with all these variables that we can extract from our data. And the plots will give you some indication of how the disease condition perform compared to the control conditions. And then you can compare drugs and see what kind of rescue you observe in these drugs. And this is just one example of one condition where we have different drugs that are able to correct or to revert some of the alterations, but not all, indicating that perhaps the better way to do would be combination of drugs or instead of a single drug. But nonetheless, it allows us to select for the drugs that are best represented to rescue or to fully rescue the condition. I think I used my time now. I'll just finish by thanking the members of my lab. So I talk about a bunch of projects, but there are two people who really push these organoid protocols forward. This is Kleber Trujillo and Priscilla Negrais. They're a couple, and both of them were really on top of the optimization for these organoid protocols. And my funding agencies and all the families that donate cells for our work. So I stop here and pass to Fabio to continue on our collaboration. Thank you.
Really outstanding. Excellent presentation and really exciting technology. So with that, I would like to introduce Dr. Fabio Tucci, who's the Chief Operating Officer and the co-founder of Epigen Biosciences. And he's going to talk about some of the development work they've been doing for several different diseases, including Fragile X. Fabio?
Thanks, Mike. I'll share my screen now, so let's see if it So hopefully y'all can see my screen now. Well, anyways, thanks Mike and Patricia for the invitation to speak in this first lecture series sponsored by FRAXA. It's a pleasure to be here and talk about one of our programs, the development of our programs towards a novel therapy for Fragile X syndrome. And I'll try to make a full screen here now, so it's a little bit better. So before I go into the project itself, I'll talk briefly about Epigen Biosciences. We are a small drug discovery incubator based in San Diego, California. We were founded in 2010 by founders that have experience in drug discovery and development and bringing compounds to the clinic. We're mostly financed by non-dilutive funding and founders funding as well. And we have a pipeline of preclinical programs ranging from lead generation all the way to IND, close to IND readiness, focused mostly on kidney, liver disease, and neuroscience. And as you can see here, the list of programs and the funding associated with which one those programs and the stages of their development. The company was founded by doctors Graham Beaton, Satish Ravula, and myself. We're three medicinal chemists by training, also drug discoverers. Hence, the company started out with a strong medicinal chemistry focus. Until very recently, Dr. Kevin Bacon was our VP of pharmacology and helped us out with a variety of assays in developing our pharmacology. Epigen relies on a variety of scientific collaborators, experts in their particular field. So in neurosciences and Fragile X and organoids, specifically Dr. Alysson Muotri, who just spoke to us. Also in Fragile X, we were collaborating with Dr. Devin Binder of UC Riverside in translational neuroscience, and Dr. Elizabeth Berry-Kravis, clinical Fragile X clinical research. And for example, in nephrology, we collaborate with Dr. Kumar Sharma of University of Texas in San Antonio, and Dr. Rohit Kohli in liver disease at the Children's Hospital of Los Angeles. So as the audience here it probably knows very well already about Fragile X syndrome and the etiology, genetics, and the molecular features of Fragile X, but I will just say a few words so I can link up our target of choice in our program to this particular disease of interest. So as you may know, Fragile X syndrome is the most common inherited cause of intellectual disability. It is a genetic disease linked to the X chromosome, which is caused by the CGG repeat expansion exceeding 200 repeats in the promoter region of the FMR1 gene. So this leads to hypermetallation and silencing and absence or reduction of its gene product, which is FMRP, the Fragile X mental retardation protein 1, which is very important. Currently, there aren't any treatments that necessarily can cure the disease, but rather treat some of the symptoms. So there is a need for developing medicines that can possibly modify this disease. Fragile X syndrome it is probably the best known brain disorder associated with aberrant protein translation as a phenotype. FMRP is our RNA binding protein, and that protein regulates the synthesis of many proteins involved in synaptic function. So, and protein synthesis is very important for synaptic plasticity and long-term memory. To that effect, if one looks at mTORC1, which is a central energy sensing center in the cell, and it has an important impact in translation control and protein synthesis through its downstream effectors 4E BP and S6K or B70S6K. So this is a critical signaling pathway in protein synthesis. As such, S6K becomes an important target in regulating aberrant protein translation in Fragile X syndrome. In very nice work done by Patricia et al., it was able to show that in FMR1 knockout mice, which is the animal model for Fragile X syndrome, phosphorylation or activity of S6K in that particular mouse model is elevated, specifically at the mTORC1 site. And as you can see here in the cartoon, the absence of FMRP, which is in the FMR1 knockout mice, leads to failed repression of protein synthesis, an enhancement of S6K1 activity, and exaggerated protein translation. There's some links to human pathology, studies using lymphocytes and brain tissue from Fragile X syndrome patients shows an upregulation of S6K phosphorylation compared to normal controls. So therefore, based on this set of evidence, we have a hypothesis that dysregulated protein synthesis is a key causal factor in Fragile X syndrome, and that inhibition of S6K with the pharmacological tool may impact in the exaggerated protein synthesis and hopefully correct and stabilize the peripheral and neurological functions seen in this condition. Also from the work done by Patricia, it was shown that in FMR1 knockout mice, phosphorylation of S6 at both sites, 240 and 244 serine, and serine 235 and 236 is elevated compared to wild type controls, as well as the phospho-ELF4B. And that a S6K1 knockout animal then shows reduced phosphorylation, obviously, in those sites, and a double knockout FMR1 S6K1 also shows the same molecular signature. Also, in this graph B here, this group was able to very nicely show that there was elevated protein translation, neuronal protein translation using a non-radioactive labeling assay. Pharmacological inhibition with S6K, small molecule S6K inhibitors is able to reverse those phenotypes in FMR1 knockout mice. So again, Patricia was able to show with two small molecule, two compounds. One, it's a Pfizer compound, which I'm going to refer to as PF compound. And another one is, you will see later in the presentation, is a Sentinel Oncology compound called FS. Both of these compounds were administered to FMR1 knockout mice. And in these two graphs you're seeing here are the effects of the PF compound given IP via intraperitoneal injection to these animals in correcting some of the social, the interaction preference in the social novelty test, and as well as correcting a behavior in the Y-maze choice arm, which shows an increased behavior flexibility with drug treatment. So the effects here are relatively, well, you can definitely see the effects. They don't seem to be very robust, but at least you can definitely see the trend in this particular study. So that made us think and start looking at S6K as a target, and we start looking at the landscape. So there aren't very many drug-like CNS penetrant and selective small molecule S6K inhibitors available. As I mentioned before, the PF compound, which the structure is shown here, has been published by Pfizer. The indications from publications is that it is not orally available, therefore it has to be given intraperitoneally to mice, and also it has limited CNS penetration. In our hands, the compound isn't a very potent inhibitor of S6K. Enzymatic IC50 is about 300 nanomolar, and cell IC50 is about 1.7 micromolar. As far as we know, 2 so-called S6K inhibitors have reached the clinic so far, and these are shown here by the Lilly compounds. They were tested for oncology indications. Both of these compounds are potent S6K inhibitors. Not much is known about their CNS penetration profile, but one of the main features of these compounds is that they're necessarily selective. And for example, the LI301 also hits AKT very potently, which is will probably be a liability for a chronic indication such as Fragile X syndrome that's has a large number of pediatric population as well. So, and finally, the FS115 compound by Sentinel Oncology, it is bioavailable and it is CNS penetrant from the publications, but the selectivity doesn't seem to be a part of this compound. So we set out to discover and engineer perhaps a new class of S6K, small molecule S6K inhibitors that can be orally bioavailable, CNS penetrant, selective, and potent. It could be used for treating Fragile X syndrome. We based our efforts on several published X-ray crystal structures of the S6 kinase bound to inhibitors to identify specific pockets where one could perhaps engineer not only potency and affinity, but also a degree of selectivity to the molecule. So as you can see here, this highlighted area on the top, this is mainly the iterative work that takes place in our labs, where we start with structure-based drug design, followed by chemical synthesis and primary testing in the S6K enzymatic assay. And that cycle repeats to identify compounds that meet the criteria for S6K enzymatic activity, IC50 less than 20 nanomolar. At that point, candidate compounds that pass this first filter will be subjected to the S6K cell-based assay, which they need to pass the second filter, at least less than 200 nanomolar based on our criteria, as well as ADME, profiling for absorption, distribution, metabolism, excretion, toxicology, in vitro assays to assess the druggability really of these molecules, followed by mouse pharmacokinetics to assess oral bioavailability and brain penetrance of these compounds. Out of this effort then, candidate molecules, fewer candidate molecules that pass all these filters then go on to perhaps be tested for efficacy in the mouse Fragile X syndrome model, in parallel be assessed in the organoid Fragile X system that Alysson was describing. And the compounds that, again, pass this filter then go on for non-GLP safety and can be nominated eventually as a development candidate. So in the next slide, I wanted to show you and to give you a little bit of a flavor for what we look for in our molecules. So we established a criteria, desirable criteria for drug-like CNS penetrant molecules. And in this case, we try to adhere to the Lipinski rules, or actually the rule of 3, or somewhere in between, where we wanted to keep the molecules small in terms of molecular weight, with control the lipophilicity by the log P, and importantly for a CNS penetrant drug, control the topological polar surface area. Our requirements for activity is in the biochemical assay, we want a molecule less than 20 nanomolar, and in cells, at least less than 200 nanomolar. There's always a little bit of a loss in potency when you go to cells. And then druggability parameters such as in vitro inhibition of the two major drug metabolizing enzymes, 3A4 and 2D6. Stability in microsomes, both in mouse and humans, indicates to us that the molecule can be bioavailable and the first pass metabolism is acceptable. HERG tells us about the risk of cardio safety and selectivity over the AGC kinase. This is a first pass for selectivity in this case. So we had an early lead, EPGN 1370, and you can see the profile here, highlighted in this reddish type color. I'm not quite sure. It's some of the profiles. So all of the other parameters seem to be acceptable except for these, where the molecule wasn't as potent in cells for S6K or for S6 inhibition, phospho-S6. And also, this molecule had a little bit of a high clearance in mice, which makes the efficacy evaluation in mice especially difficult. What was good about this molecule is that it was selective against the AGC kinases, was bioavailable, and had good brain penetrance. So that served as our optimization starting point here, where we ended up arriving at EPGN 2036. This compound now is more potent, both enzymatically and in cells for S6 inhibition, S6K inhibition. No drug-drug interaction issues with CYP inhibition. Stability in microsomes now at an acceptable intrinsic clearance for both mice and humans, and a reasonable IC50 for HERG, selectivity, and pharmacokinetics. You will see in the next few slides, I'll show you some of the in vivo efficacy slides. And for a company small company like us, it is important for us that the compounds are novel and can be patented so that we can eventually commercialize this technology. So this is the case here. Before I go into the efficacy slides, I want to show you the pharmacokinetics in mice, given both at 30 and 100 milligrams per kilogram orally in this case. So you can see each of these graphs has 3 individual mice. So these are the different lines that you see. So you can see the mean values in the table below, where at 30 mg per kg, one gets an AUC of about 4,900. And at 100 mg per kg, you get an AUC of about 20,000, I would say. The brain to plasma ratio, the BP, at 4-hour time points at 30 mg per kg is about 1.9, close to 2. So that would translate to about 1.3 micromolar total concentration in the brain. And at 100 milligrams per kilogram, the brain to plasma ratio is 4 hours is 2.6, is a little bit higher, with the with the total concentration of 7.6 micromolar. So way well above the cell IC50 of the molecule here. Most importantly, we can see that both of these doses, we have sufficient coverage of the enzyme over the 24-hour period based on its IC50. In addition to the AGC selectivity panel, we ran a more extensive kinase profile selectivity panel from Eurofins Discovery for 123 targets using the gold standard radiometric assay. And in addition to S6K itself, which showed up in this particular screen, there are 4 other targets that were hit at less than 50% at 1 micromolar concentration. The IC50s are seen here. The 2 kinases that show a little bit closer in terms of selectivity, BIKE and NEC9. BIKE is known to play a role in skeletal development and patterning. And And we actually don't necessarily know yet of the impact of these particular kinases, but it's clear that given the difference in fold selectivity over IC50, is that the cell IC50s for these, just like S6A, is likely to be much higher. So we hopefully don't think that this would be too much of an issue. So in this slide here was our first efficacy measurement, which was done in the laboratories of Dr. Xinyu Zhao at University of Wisconsin-Madison. She's here attending the talk today, and we're very thankful for the collaboration with her lab. So in her lab, they injected EPGN-2036 at 30 milligrams per kilogram intraperitoneally. And you were able to see here with the 4 different behaviors that there was a rescuing hyperactivity, the open field, social spatial learning, social interaction, and social novelty deficits that were corrected or rescued by the compound. What was also very interesting in this particular study is that we collected a hippocampus of these animals, and we were able to show that first, the phospho-S6 is elevated in the FMR1 knockout mice in comparison to wild type, in agreement with the Bhattacharya paper, as well as compound treatment, in this case, led to a reduction in phospho-S6 in the hippocampus. So that's a suggestion of target engagement in the brain, or in the hippocampus, at least. We also tested the efficacy, in vivo efficacy of EPGN-2036 via oral delivery in a mouse FMR1 knockout 2 mice. And this work was done in collaboration with Dr. Patricia Cogram of FRAXA and Mike Tranfaglia as well. We're very grateful for the collaboration, for the opportunity to obtain this kind of data. In this case, EPGN-2036 was dosed orally at 4 different doses. In this case, 10, 30, 60, and 100 milligrams per kilogram once a day for 3 days before behavior measurements. And what we can see here is there is a dose-dependent normalization, I guess, of aberrant behavior in all of these particular behaviors, such as an open field and sociability, fear conditioning, hyponeophagia, and the nesting behavior. So hyponeophagia, I think it's a very interesting measure of anxiety, I guess, of these particular mice, and the open field and sociability also seem to match what Xinyu's lab had seen. So in this particular case, a dose of 30 mg per kg was considered to be the MED, the minimum efficacious dose here. 10 milligrams per kilogram, there wasn't much of an effect, but at 30, we start to see an effect. So this is based on the pharmacokinetic profile, then we can definitely project out the exposures for this particular doses. So once we had the efficacy established for EPGN-2036, one of the big questions was, is a kinase inhibitor safe to be administered chronically, right? So I think the first step for us was a 14-day non-GLP rat screening safety study, dose range finding study, where we gave this compound at 3 different doses, 100, 300, and 1,000 milligrams per kilogram once daily to female SD rats for 14 days. And the results, the preliminary results at least, show that there's no drug-associated mortality with this particular drug for 14 days in these rats. We did see, though, however, a dose-dependent reduction in body weight gain. You can see the graph here where There's more observable effects at 1,000 and 300 milligrams per kilogram, whereas the 100 milligram per kilogram dose mirror pretty much the vehicle controls. So that suggests some type of tolerability at a higher doses for this particular compound. And therefore, we assign, based on some of the other parameters, an NOAL, no adverse event effect level of 100 milligrams per kilogram to this particular, in this particular study. TK results are shown here. So these are essentially the exposures seen at both day 1 and day 14 for the 3 doses in this study. The key conclusions here is that, well, which is important. One is the exposure increases with dose, not in a proportional way. In general, exposures at day 14 are higher than day 1. And we observed that females have higher exposure than male. And that's the reason why we use females in the safety study to maximize exposure based on the dose here. In terms of safety margin, considering a NOAL, preliminary NOAL of 100 mg per kg and a mouse oral efficacy of 30 mg per kg, based on the calculations, we can essentially come to a safety margin close to 15-fold, which may be acceptable for advancing this compound to human studies for this particular indication. So we're aware that many compounds and many mechanisms of action have been tested in behavioral mouse models of Fragile X syndrome, but eventually failed in the clinic. So what can we do here and try to mitigate that possibility and try to find early whether we have a chance of being effective in people? So one of the recommendations that came out of Dr. Barry Kravis' work was that one would try to do preclinical work in which comparable biomarkers between the animal and the human could be evaluated as a way to try to increase the translation potential of the study. So based on that, we teamed up with the Binder Lab at UC Riverside. They've developed a mouse multi-electrode array for measuring EEG in FMR1 knockout mice, and based on several of the parameters, they were able to see that these are remarkably similar to what's observed in humans with Fragile X syndrome. So that gives us potentially a translatable biomarker in which one can early measure the efficacy or at least the target engagement of the compound. And this is my last science slide. We're working with Alysson's lab, and as he showed in his work, they've been developing organoid systems from, in this particular case, Fragile X syndrome patients. And the system is being characterized. For example, you see here on the left-hand side panel, two of the pictures. One is a control normal organoid, and the FX is the Fragile X organoid. So you see clear differences in organization. The Fragile X is less organized. And some of the molecular characterization, it's also consistent. So for example, panel B, there's an absence of the FMRP protein in the Fragile X mini brain. And the synapsin and PSD95 are reduced, which is a sign of reduced synaptogenesis, which is a feature of Fragile X. And also, most importantly, there is an increase in S6 phosphorylation in these organoids. On the right-hand side, this is a fresh off the press slide where Alysson treated an early organoid, I guess, with the 5 micromolar EPGN 2036 and recorded the electric current of these organoids. And after 1 week treatment, there's a decrease in hyperexcitability of these organoids when treated with a compound when compared to control. So that could be a good sign. I think more characterization is needed, and this work is underway. As a summary, I hope I convinced you that S6K is potentially a disease-modifying target for treating Fragile X syndrome. Epigen has developed small molecules for inhibitors of S6K, which are potent in cells, selective, and capable of penetrating the brain. We've been able to establish behavioral efficacy preclinically. We have an acceptable safety profile and characterization of this molecule together with FXS brain organoids, as well as EEG translational biomarker development is in progress. We hope to report those in due course. And finally, I would just like to thank quite a bunch of people. I think the Epigen team is listed here: Graham, Satish, Kevin, Chandra, and Sukhjun. Thanks for your effort. At UCSD, Alyson's group, Isabella, Spencer, and Wei Wu were very important in getting this work going. I'm very thankful for Xinyu Zhao and Minji Shen at University of Wisconsin-Madison. Especially thankful to Mike and Patricia at FRAXA for supporting the work so far. We've done the PK and the safety work at Quintara and Calvert. And finally, I wanted to thank NIMH at the NIH for funding this work so far. And this concludes my presentation. Thank you.
Really great, a wonderful demonstration of collaboration between academia and industry. I think that's particularly important in Fragile X. And we have some time, plenty of time actually, for questions. And we have a couple that have already come in. From the this is from the first presentation. 4-month organoid graph. What cells are green and pink? Are they intermediates between specific cell types?
That's correct. Yeah. So the ones that were not labeled are cells that are intermediated progenitor cells, where the identity of the cells cannot be fully defined by the single cell analysis. As I said, as they mature, the level or the number of intermediated progenitor cells become less and less, and the cells become more mature, acquiring their identity.
And from Xinyu Zhao, Alysson presented MEA data.
Yeah, that's for me as well. Yeah, so it's a matter of cost to perform the MEA in a high throughput. It's more costly than the imaging-based electrophysiology with calcium imaging. That's the only reason we want to move to the MEAs in a high throughput fashion. It's just a little bit more challenge, and the cost is higher. So the next one is also for me on the 2D versus 3D organoids. Right now, what I presented is a flat, a planar EEG where the organoids are sitting on top. So although the organoid remains on a three-dimensional structure, the recordings are actually happening in 2D. So now we have other MEAs that can surround the organoid, so the recording can be done in 3D as well.
That's interesting. And I had a question related to that as well. If you take the same organoid that you've recorded on a 2D multi-electrode array, or microelectrode array, excuse me, and you just flip it around, let's just say you roll it around to the other side, do you get fairly consistent results from one surface of the organoid to another?
We haven't done that specific experiment, so I don't know. I guess we will know with the 3D organoids if the activity is similar all over or not. Since each organoid has a slightly different cytoarchitecture, I think we're gonna see like some subtle differences. So the 3D MEAs might help us on finding those differences or averaging the organoid. Another related experiment that we did was to get a 3D organoid and to flatten that 3D organoid. So we dissociate the organoid and replate in a 2D monolayer. And then the complexity of the network is gone. So you really need that three-dimensional structure to be able to capture those neural oscillations and to evolve over time.
Okay, so the next question from David Walker is about the aqueous solubility of epigen 2036.
I can take that one. The solubility, we haven't really formally measured the thermodynamic solubility of 2036. We do have to formulate this compound. For example, if you want to do an IV injection, for example, and obtain a solution, you do need to make a solution with solutol, for example, to aid solubilization. So it isn't one of these things that is freely water soluble, or 1 or 2 mg per mil, but it's more like in the micromolar range. I mean, the PK studies, in vivo studies, and safety studies, they're all done using suspension formulations, very standard, typical way, and we do see great absorption and oral bioavailability. So that's what we know.
Okay, and Milos asks if you saw any cell-specific phenotypes in the Fragile X organoids. And I guess I would elaborate on that and say, so there's structural phenotypes, morphological phenotypes, synaptic phenotypes, and are there actual electrophysiologic phenotypes in individual cells?
Yeah, so for the structural and cellular phenotype, we're still characterizing. It seems like some of the populations might be slightly off on the Fragile X organoids, but this remains as a preliminary data. We do see gene expression alterations. We do see alterations in the number of excitatory synapses. In electrophysiology, on patch clumping, we see hyperexcitability on cortical neurons. So we want to measure these neural oscillations that will require the organoids to mature for at least 6 to 8 months. But that's the ultimate goal, is to test drugs, including Fabio's drugs, on the ability to rescue these neural oscillations. I think that's the gold standard here.
Okay, and David has another question. Looks like drug development for you, Fabio. What does CPD look like in CERAP or similar safety panel?
So we really haven't done an expanded CERAP panel, like a safety 44 or 70, safety 70, I think 7 or 78. It's a group of targets, I guess, the CERAP puts together that you probably don't want to hit. So we haven't really done those yet. What we've done was the expanded kinase panel, which to us was most important in the beginning, given that a lot of kinase inhibitors have a difficult time in being selective over other kinases, especially kinases of the same family. So we were able to do that and demonstrate reasonable safety. The second part of the question is wondering if any other clinical signs in the rat safety study. So it's still preliminary. I think the results, clinical chemistry, didn't really see necessarily anything too dose dependent or anything like that we can point a finger to. Pathology and all those things is still a little bit pending, so we need to see. But in terms of clinical signs, yes, body weight gain, and the high dose group, the 1,000 mg per kg, the animals weren't looking too happy, and it's probably understandable. So.
And Patrick McCampbell at the Bear Lab at MIT. Hi, Patrick. asks if the change in hyperexcitability is amplitude or is it also frequency? I get the sense it's very preliminary, but maybe you could guess.
It is very preliminary, but it seems like we rescue both the amplitude spiking and the frequency of events. So we're quite excited with this preliminary data. Looks quite promising. I think is one of the robust rescue phenotypes that we had in a long time in neurons. So this looks promising. Looking forward to see how this looks in the organoids.
Osman asks if there's any drug in clinical use based on testing in organoids.
Yes. Well, I mean, in clinical trials, the data from the Icardi Gutierrez, we published that in 2017. In 2018, the clinical trial started and has been going on since then.
Judith asks, is the compound inhibition of S6 kinase selective for neurons or any cell type?
As far as we can tell, is any cell type. So the compound will inhibit S6K in peripherally as well as centrally.
And it's quite ubiquitous as an enzyme.
Correct.
From Raya, how is MEA of the organoids compared to brain activity of individuals who are several years or several decades old?
Yeah, so the organoids really mimics these early stages of neurodevelopment. It might be that there is a rescue of activity that is kept in a more adult brain. We don't know. The idea to compare with kids is our next step. So that's what we are doing with autism. But we are also are trying to age these networks to resemble more old networks or networks from an older brain, even though the age of the organoid remains young. And we are doing that through stimulation.
And could I just ask, what's the current Guinness Book of World Records title for the oldest organoid?
I don't know. I mean, it seems like you can keep them for several years. I think our record is like 3 years. But the electrical activity plateaus after a year or so. So we don't see that much advantage of keeping them for longer periods of time, unless we can continue to increase the activity or maturing these networks. That's exactly what we are doing with the stimulation part.
Osman asks, how specific is the test for S6 kinase in comparison to other kinases, and how many can be tested? I can already imagine the sky's the limit depending on how much you want to spend on that, but would you like to comment on that, Fabio?
Yeah, you hit the nail on the head, Mike. No, the assay, the biochemical assay is very specific. I mean, it's a recombinant enzyme assay, so you can, and it's actually not that expensive. We do that at Reaction Biology. I can say the name here, the company, and it's fairly robust. So what's a little bit more expensive is the cell-based assay. So throughput for the biochemical assay, yeah, it can be very high, I think. Cell-based maybe a little bit lower.
Another question from David Walker. Hi, David. Are there plans to look at tox in large species like dogs? I would imagine.
So yes, eventually as this compound progresses, or any other compound in this series progresses towards development, one would have to test, at least for safety, in a non-rodent species. I think what's going to dictate whether it's dog or whether it's monkey, it's the metabolite profile of the compound. So we will have to do a metabolite identification test to see, if the metabolites generated in humans is similar to the metabolites generated in dogs or monkeys as well as rats. So you can tie the safety of your compound as well as metabolites and translate that into the clinic. So yes, there is a plan. We're not quite there yet.
And Shika is wondering about testicular cells as a model for Fragile X. especially since FMRP is expressed highly in testicular cells, and there are phenotypes there in humans. Has anyone looked at that?
I think it might be like a good model for kinase activity or even like some target gene expression. But definitely, I mean, you cannot measure electrophysiology or number of synapses, things like that. That's always the balance between using the relevant cell type and other cell types that you might be able to do it in a high throughput fashion. But really depends on your assay.
I think also the high expression in testes may be an artifact of just the high turnover in cells in that part of the body. I mean, you tend to see more FMRP expression in cells that are turning over more rapidly in the body. Osman again, how much, basically asking about the percentage of the phenotype. How much is S6 kinase contributing to the Fragile X human phenotype and pathophysiology? Do you have organoids from Fragile X patients with no clinical symptoms? If I'm understanding what you've done so far, your organoids are generated from normal cells by knocking out FMRP. Is that correct? Or are you actually using patient-derived?
We have both. We have from patient cells and isogenic cells by knocking down the gene in a control cell line. But we don't have like an extensive number of patients. So we don't have Fragile X patients with no clinical symptoms, for example. And I don't know if this strategy will work. Once you reprogram the cells, the methylation is fully erased. So you have to assume that once you differentiate the cells, you're going to recapitulate all the epigenetic marks, which might not be the case. So yeah, but definitely, I mean, it's worth a try. And I really don't know how much of the S6K contributes to the Fragile X phenotype. Fabio, remember like some papers here, right? I mean, on rescuing some of the phenotypes, specifically targeting that kinase, no?
Yes, as I referenced, I showed some work that was done establishing S6K as a viable target for Fragile X syndrome. I mean, the reason why S6K is attractive to us, at least, is because it's in that mTOR, PI3 kinase mTOR pathway, which is many kinases. They're very important for many other cell functions, and targeting S6K, which is the terminal kinase here, probably the potential for deleterious side effects is probably lower. So I think targeting the entire pathway, I think it's a very promising strategy. It depends where you're going to target to, balance the benefit with the risk. At least that's my thinking. So
that seems to be the most common question we get about every single therapeutic target is what percentage of the overall phenotype comes from this particular therapeutic target, and it's really impossible to say. And we went through that with mGluR5, and nobody really got to a conclusive answer. And I don't think it is necessarily possible to understand that until you get to clinical trials. Milos is asking how much the necrotic center of the organoids influences the microelectrode recordings in aged organoids, and how necrotic does it get, and how long does that take, I guess I would add.
Yeah, so you at least in our hands, using our protocol, we start seeing some necrosis after 6 or 8 months. We don't see that influences the NEA, again, because we are recording from the outside, and even in age organoids, like a year or so, we don't see that the activity goes down. It plateaus, which I think is what would expect by not maturing more those neurons or not adding more neurons to the network, but we never see it going down. So I think as long as you keep recording from the neurons in the outside, the networks that they can form in the cortical plate continues to be active. So it doesn't seem to influence that much.
Vanessa asks about glial cell marker expression in the organoids.
Yeah, so the organoids, once they get about 4 months of age, you start to see some of the astrocytes being present, and the population of astrocytes will continue to increase up to 9, 10 months, reaching 50% population inside the organoids. So you start with 100% neuroprogenitor cells and neurons, and after 1 year, you have 1-to-1, one astrocyte to one organoid. And microglia cells, depending on the question, can be infused or co-cultured with these organoids. So you have to, because they have a different embryonic orange, you grow them in parallel and then you add them on the organoids. But we cannot create an organoid with the microglia already there because we patterned the organoid early on.
And Raya asks how you get oxygen to the deeper parts of the organoids, and that there is a recent paper suggesting that hypoxic stress can skew the neuron types in organoids. Have you figured out a trick to get oxygen in there?
No, our organoids are also not vascularized. This is something that we are teaming up with the bioengineers here to vascularize these organoids. So we hope that this will not be a problem. We don't see as much as a problem as it was previously reported. So there are variability on how the different labs grow these organoids or how they, which protocol they use. So these are all things that you need to take in consideration. As I said in the beginning, we spend lots of time optimizing the culture conditions. So maybe that's why we see less of a problem here.
Shika asks if organoids are acceptable for presenting data to the FDA, and how are organoids standardized across multiple experiments? What criteria are measured for controls and reduction of noise?
I definitely see a tendency on the FDA to start looking at organoid data. I'm not sure if we're going to completely replace the animal. Well, I mean, you can't replace for toxicology, for example, but as an efficacy, I think would be like a complementary assay moving forward. They standardize across multiple experiments. Yeah, you have to show, I mean, how robust or reproducible is your assay. So that's why we try to use multiple organoids, differentiated from different people, as well as isogenic cells. And you have to show your variability. And of course, I mean, the drug to move forward, the rescue should be higher than the variability that you have in your assay. A big surprise to us in this area is what we've been used in the past, monolayers of neurons. The variability there is higher than in the organoids. So we are quite happy with the variability that we see in the organoids. Seems like much reduced compared to the traditional monolayer culture of neurons. Which is a plus for us.
Julie asks, mTORC inhibition doesn't seem to be beneficial in Fragile X mouse models. So how does this fit with benefits from S6 kinase inhibition?
Well, I think mTORC1 inhibition directly, I think that would have to be done with rapamycin, for example, something like that. And I probably have to go take a look at some of the papers and see, but I imagine that it would be difficult to do that in the context of Fragile X. I know that some PI3 kinase inhibitors, different small molecule PI3 kinase inhibitors, which is upstream of mTORC1, for example, they seem to be effective in FMR1 knockout mouse models as for behavior, as far as I can remember. So yeah, so I don't really know, maybe perhaps rapamycin is not the ideal compound with TORC1 inhibition or something like that, but we clearly see the beneficial effects with inhibiting the downstream effector S6K in this case.
David asks, in the in vivo studies, were all the mice males, and what age? Wondering if the compound could be efficacious in both young and aged mice.
I think the compounds were a mixed males and females based on the protocol. I don't know if Patricia is online to be able to just clarify that for us, but
the mice were all males in our case. I don't know in your other studies because there were other studies doing
if those were all, yeah.
So in our studies, they were all males, 2 months more or less were the age. I don't know in the other study.
yeah, the other one, Zinyu is mentioning it's 2 or 3 months old males as well.
Melanie asks if you get any ventricles in your organoids.
Yeah, I think I had a picture over there. It's a beautiful ventricle surrounding with the progenitor cells and the radioglia cells. The anatomy of the organoid is quite beautiful. I like that.
And Shika asks, testicular cells high FMRP production results in enlarged testis as a phenotype. I was curious if simply using drugs to recover that phenotype in Fragile X boys will be a safer alternative in terms of dosing, screening, efficacy, et cetera, before going from animal models to human brains. Would love to talk to anyone who has thoughts about this.
Mm-hmm.
I'd be happy to talk to you about that anytime, Shika. Ah, looks like we've finally gotten to the end of the list. Lots of good questions.
Well, I'd just like to add, this is Katie Clapp from FRAXA. I'd just like to add thank you to Fabio and to Alysson and everybody who's attended. This was the first of our series, and we plan to do the next one soon.
And thanks to Patricia for actually coming up with the idea. It was actually all her idea originally.
Yes.
There was one more question from David Walker. It says, are there any plans to publish on the chemistry?
Yes, we first need to file. We're in the process of filing patent applications first, so before we can publish on the chemistry. So that's the main reason why we can't quite be so upfront about structures and things like that. But that's going to happen in due course. It's part of the process.
Great. Well, thank you guys. We're right on schedule. Perfect. Looking forward to the next one. Hope you can all join us then.
Thank you, Michael. Thank you, Patricia. Thank you, Fabio.
Thanks,
Great
Alysson.
questions. Thank you.
Check the FRAXA website for upcoming events.
All right. Bye.
Bye.
Bye-bye.